Today we will talk about the nephron, which is the structural and functional unit of the kidney. In this lecture, we will discuss the structure of the nephron. In the coming lectures, we will also discuss the functions of the nephron. Now, as we have previously discussed that the kidney is made of around 1 million nephron, and the number of the nephrons is fixed throughout the life. But after reaching the age of 40, there is a 10% decrease every 10 years, and after reaching around 80 years, there are about half non-functional nephrons. But they are sufficient at that age due to their adaptation. Now, let's talk about the structure of the nephron. As we previously discussed that the pyramid, the renal pyramid is made of nephrons. Now, we will take just one nephron and we will enlarge it under the microscope and see that what are basically the different parts of the nephron. Now, to start, the nephron initially has a renal corpuscle which is made of a glomerulus and the Bowman's capsule. The glomerulus is basically a bunch of capillaries. So here is the glomerulus. This red color is the glomerulus, which is basically made of capillaries, and through the glomerulus, blood from the capillaries is filtered into the Bowman's capsule. Now, fluid is filtered from the glomerulus into the Bowman's capsule, and it will now continue to move in these tubules. The initial portion of this nephron tubule is known as the proximal convoluted tubule. As we have discussed that there is a cortex, the outer portion, and a medulla in the kidney. This red color is the cortex. These pyramids are present in the medulla. So similarly, some parts of this nephron will be present in the cortex, and some of the parts of nephron are present in the medulla. So the initial part, the proximal convoluted tubule, which is after the Bowman's capsule, is basically a cortical structure. It is mostly present in the cortex. After the proximal convoluted tubule, we have the loop of Henle. Now, the loop of Henle has two limbs, the descending limb and the ascending limb. This is the ascending limb, and this one is the ascending limb. So the descending limb is basically coming down, the ascending limb is basically moving up. Now, each limb, the descending and the ascending, here have some thick and thin portions. This descending limb have thick portion known as thick portion of the descending limb of loop of Henle. This portion is the thin part, and it is known as the thin segment of the descending limb of loop of Henle. Then we have the ascending limb, and in the ascending limb as well, we have the thin segment of the ascending limb of loop of Henle, and finally thick segment of the ascending limb of loop of Henle. Then we have a dark colored area, this black colored area, which is known as the macula densa. Macula densa is important in sensing the nutrients, the concentration of different nutrients in the tubule, and is important in controlling the different nutrients and electrolytes. We will discuss it in detail in the coming lectures. So, initially the glomerulus, then the Bowman's capsule, then the proximal convoluted tubule, then the thick and thin segments of the ascending and descending limb of loop of Henle. Finally, after the macula densa, we have the distal convoluted tubule. And after the distal convoluted tubule, we have the connecting tubule. Now, this connecting tubule is connecting this nephron with the cortical collecting tubule. So, a lot of connecting tubules, they make the cortical collecting tubule. So, this is one nephron. Here we have another nephron, and both the connecting tubule of both these nephrons are basically connecting these nephrons with the cortical collecting tubule, or a lot of connecting tubules then make a cortical collecting tubule. Now, this portion of the nephron, the proximal, the distal, and these portions are basically present in the cortex. That's why this is known as the cortical collecting tubule. Then the cortical collecting tubule, it enlarges in size, and it basically becomes the cortical collecting duct. It becomes the cortical collecting ducts. So it becomes the cortical collecting ducts, but it is still in the cortex. This is basically cortex. This region is the cortex, is the medulla. So this is the cortex. This is the cortex. Here is the medulla, and we have basically taken one nephron from here and we have enlarged it. So here we have the cortex. This line basically divides it. So some portions are in the cortex and some are in the medulla. If we talk about this nephron, it is basically present, its more structure are present in the cortex. That is another thing which we will discuss in the coming lectures. But if we specifically talk about this nephron, these proximal and distal tubules are present in the cortex, and these loop of Henle, especially the thin segments, they are present in the medulla. So the distal convoluted tubule is also present in the cortex, and it is a cortical structure. They become the connecting tubule, then the cortical collecting tubule, and finally becoming the cortical collecting duct. Here it becomes the cortical collecting duct. Now 8 to 10 cortical collecting ducts 8 to 10 cortical collecting ducts, they join together and form a larger collecting duct that run downward into the medulla and becomes the medullary collecting duct. They 8 to 10 cortical collecting ducts join together and they run deep into the medulla and becoming the medullary collecting duct. And finally, the collecting ducts, these all collecting ducts, they merge together and they open into the papilla. Now we know that this is the papilla, this portion is the papilla. So all these larger collecting ducts, they open at this point into the papilla, and then we know these papilla basically open into the minor calyces basically open into the major calyces into the pelvis, and pelvis into the ureter, from ureter urine go into the bladder and then out of the body. So this is how we basically enlarge this one nephron from this pyramid and the kidney, and then at the same time we will try to look at this nephron in this portion of the kidney. Like we have to understand the structure of the nephron, and at the same time we have to focus that which part of the nephron is present in the cortex and which part is present in the medulla and how it is correlating with this kidney. So this is something which we have discussed previously in detail. Now to summarize this, the initial portion of the nephron, which is the renal corpuscle, is made of Bowman's capsule, and the glomerulus fluid enters into the initial portion of the tubule known as the proximal convoluted tubule, then into the thick segment of the descending limb of loop of Henle, then the thin segment of descending limb of loop of Henle, then thin segment of ascending limb of loop of Henle, then thick segment of ascending limb of loop of Henle. Then there is macula densa, and after the macula densa we have the distal convoluted tubule. After the distal convoluted tubule we have the connecting tubule. This is the connecting tubule. Then the connecting tubule join the cortical collecting tubule. The cortical collecting tubule, they basically come down and they enlarges and become cortical collecting duct, but they are still in the cortex. These cortical collecting ducts, around 8 to 10 cortical collecting ducts, they join together and they come down into the medulla and become the medullary collecting duct. And finally, the medullary collecting ducts, they open into the renal papilla, or this part of the kidney at the tip of the pyramid. Now we must know that there are around 250 these large ducts, 250 large ducts, and each duct is basically taking urine or waste from around 4,000 nephrons, or each duct is basically connected with around 44,000 nephrons. So each kidney has 250 large ducts, and each duct is basically taking or it is connected with around 4,000 nephrons. So that's all about the structure of nephron. In the next lecture, we will discuss the differences in the nephron structure, the cortical and the juxtamedullary nephrons. So thanks a lot for watching the video.